I'm Dr. Aidoo with the Department of Anatomy and Cell Biology, School of Medical Sciences, University of Cape Coast. I present to you histopathology tissue processing. So before you can observe and view any tissue which has been put on a slide, these are the processes that the tissues go through. We call that tissue processing. So you take a structure like the liver, then you have to cut, okay, with a surgical blade. Then you fix, okay, or fixation. The importance of fixation is to achieve the 3 Ds, that is decomposition, digestion, and drying or dehydration. The importance of this fixation to achieve these 3 Ds is by cross-linking proteins, and by way of that, it's going to preserve the structures. Examples of some of the fixatives are formaldehyde, glutaraldehyde, osmium tetraoxide, methanol, ethanol, etc. Then the third stage will be to dehydrate in graded alcohol to absolute alcohol. So you have the graded alcohol in different percentages, as you see. So you can have like 70%, 80%, 90%, 100%, okay, in that order. Then you come to infiltration. I've deliberately ignored the clearing because I want to do some connection here. So you need to infiltrate the tissue with wax or paraffin. But because paraffin or wax is not miscible with alcohol, you first have to clear the alcohol with xylene. So xylene is miscible with alcohol and it's also miscible with wax. So it serves like an interface between the two. So we call that clearing with xylene. So that is clearing. Then obviously the infiltration will now come in. Then you embed also in wax. Okay, so this is the embedding. Embedding is to give it some body, the structure some body or weight or covering. For the tissue is quite small, so you need to hold it within a body. Okay, and the wax plays that role. And you realize that when you are doing the sectioning with the microtome, you need to block the tissue and put it in a tissue holder, and you would need the embedding structure here to help position the tissue well in the microtome. Then you section with the microtome, obviously, into thin slices, okay? Very, very thin slices, sometimes even in micrometers, okay? Then you stain with dyes. The simple ones are the hematoxylin and then eosin. So these are basic steps in tissue processing. It's more complex than what you see here. This is just a simplified form. Now with the fixation, you can have two main types. You have what we call the immersion fixation and then perfusion fixation. The immersion fixation is where you have your fixative, as we've mentioned, like the formalin or methanol or whatever, and then you put the structure or the tissue that you want to fix in it. All right. That is just called immersion fixation because you are immersing the tissue into the fixative. Then you also have perfusion fixation. The perfusion fixation, you make an entrance with a needle here in the left ventricle of the animal that you want to fix. Okay. In that case, the heart should still be beating. So you put the animal in a semi-active stage. Okay. You can chloroform it, and then the heart will still be beating. Then you introduce the fixative with a needle into the left ventricle, and then you cut the right atrium, a portion of it. So once the heart is beating, it's going to send the fixative into general circulation, just like in the ordinary circulation, okay, where you have oxygenated blood and it's being taken to systemic. Once you put the fixative in the left ventricle, it's going to be pushed through the aorta and then be sent to all the parts of the body. And you know that once oxygenated blood is sent, it comes back into the right portion of the heart as deoxygenated blood. In this same way, the fixative is going to travel through all the tissue, and then the exudate is going to come into the right chamber of the heart, and then it will be pumped out. All right. So that is what happens. So this is just a diagram to illustrate what I just said. So you introduce this. So you have the fixative here in yellow. That is a saline together. And then you put it in the left ventricle. Then it goes into circulation, okay, and then comes here, and then the right ventricle pumps it. To take it out because you have cut a portion of the right atrium. So when the right ventricle contracts, not going to push the substance into the pulmonary circulation. No, because of the cut, it will exude out. Right? So that is what is termed perfusion fixation. Now, I mentioned that during the dehydration, you put the tissue in graded alcohol, which are different percentages of alcohol from the lowest to the highest percentage. So, have 50%, 70%, 90%, 100, and can even repeat the 100% to ensure that you have actually done a very good dehydration. Then that is the xylene, okay? Then that is paraffin. So, alcohol replaces water in the cells. That is the dehydration. Xylene dissolves alcohol, and paraffin displaces xylene. Specimen is now ready to be embedded. So if it's a skin tissue, for instance, you cut it into small pieces, you put it in this container called the cassette, and then you put the cassette in the tissue processor if you have one. So the tissue processor will contain the graded alcohol, okay, and the xylene as well. And to do all that and then bring the tissue out, then you do the embedding with the paraffin, and then you block the tissue, then you put it in the microscope and you section. Okay, once you section it, will come out as a ribbon, very flat, thin slices, and then you put them in on top of the water and skillfully pick it onto the slide, and you mount it, and then you stain and cover it with a cover slip. So for this brain tissue, it's being put in this container that is immersion fixation, but then obviously they did a cardiac perfusion before they put it in this. So this is A, cardiac perfusion, obtaining the whole organ fixation in 4% paraformaldehyde, then the resin in phosphate buffer saline. You dehydrate, you clear, you wax immerse, you embed, you trim, you section, then you collect your specimen. So that is the process it is going through: fixation, rinsing, dehydration, clearing with the xylene or whatever substance you use for the clearing, then tissue embedding. Then you have these sections. These planes are just examples of what the sections that the microtome has cut the tissue into. So you have this summary tissue sections placed in label cassettes like that, tissue processing in this automated tissue processor. So all these jars will contain solutions, okay, where the tissues will be put in them automatically per the time you have set. So for instance, if you want the tissue to be in 60% alcohol for 1 minute, after the 1 minute, it will rotate and then take the tissue to the next stage of, let's say, 70%. Then after another duration that you have set, whether it's still a minute or 2 minutes or whatever, once that time elapses, the machine will rotate and then put the tissue in the next solution until it is done. Then you do the embedding with the wax, the microtome. So you fix the blocked tissue in there, and then you operate the microtome. Then you pick the ribbons onto water, and then you skillfully put them on the slide. Then you stain. This is an automated staining machine which will contain your stain of interest. So if it is H&E, hematoxylin, and eosin, it will be there. Then it will do the stain. So the microtome is just like the sewing machine of the tailors or seamstresses. So it has this handle where you wind around, and that is where you put the tissue which has been embedded, okay, or blocked. So that is where it is. That is a tray. That is a knife holder. You put the knife inside there, and as you wind this, it will be cutting. So this is how it is. That is the tissue block. So you can see within it some two tissues, okay, like a half moon. Yeah. Then the blade will be there, and once you wind it, and you have actually determined the thickness, okay, that you want to cut by positioning the blade, okay, and then also the block tissue at a particular distance, which will specifically cut it as you wind it at that particular thickness you want. So once you stain, you are getting close to the end of the whole journey. So basic stains and then acidic stains are the two major stains that we use. So an example of the basic stain is the hematoxylin, toluidine blue, Asian blue, methylene blue, and these will stain blue or blue-black. And acidic stains, example is the eosin, to stain purple or pink, and sometimes red. So which structures are going to be stained by the hematoxylin and which ones are supposed to be stained by the eosin? It depends on the biochemical property of the structure. So basophilic structures are negatively charged and they are acidic substances. So example, the nucleic acids, glycosaminoglycans, glycans, they have high affinity for basic dyes. Okay, so they will pick the basic dye, which is the hematoxylin. And if you know the color of the hematoxylin dye is supposed to be blue or blue-black, then anytime you observe an H&E micrograph and you have blue or blue-black colors there, then that means that they have been stained, accordingly by the basic stain hematoxylin. Then you have acidophilic structures. They love the acid type of stain. They are positively charged. And then examples are proteins like mitochondria, collagen. They have high affinity for the acidic dyes. Okay, so because they are positively charged, they will be attracted by negative dyes. Then you also have what we call special stains. So you can see that you have different colors appearing. This is a neuron with dendrites and axons. So you have different stains per what you want to achieve. Okay, so you have stains like the Giemsa stain, which is used to stain malaria parasites, like in this blood film. Okay, you can have that. You also have the trichrome stain and a whole lot. So these are just examples of other stains. So this H&E. Periodic acid shift is also a type of special stain. Goblet cells are usually not stained with H&E, so this is a special stain giving the goblet cell color. So you can tell that all these blue or blue-black structures are actually nuclei, okay, because they have picked the hematoxylin. And then portions that are looking pinkish in color are all eosin stained regions. So when I have this, you can tell, okay, very pinkish. And then you also have dark stained regions. All will be nuclei. This is actually the retina. So you are looking at the cell bodies of the bipolar neurons and then the ganglionic cell layers as well. And that is the choroid, that is the sclera. Okay, so you can see very nice, arrangement, dark and light. So the dark ones are hematoxylin, the light ones are eosin. You can see that very pinkish in color. It's also a portion of the retina and the optic nerves that are traveling. So this is the bronchiole. That is, you are looking at the respiratory system now. Again, you can see very pink outline here. It's smooth muscles. That is the lining epithelium. And this is the pituitary. So anterior lobe, posterior lobe, all H and E stains. Very beautiful. So trachealis muscle. So we are still looking at the trachea respiratory system. You can see the pinkish nature. And then you can also see some basophilic, or hematoxylin color, right? Blue and blue-black. Can also see some of the blue and blue-black within this respiratory epithelium because there are nuclei there. So again, you can see pink colors there. You can see dark region interspersed. This is cartilage with the chondrocytes there. These are smooth muscles, trachealis muscle. Part of the pituitary too, so you can see basophils there, acidophils, or they are picking more of the eosin. This is picking more of the hematoxylin. So you can see that it's dark, and this is more pinkish. These are sinusoids with blood vessels. It's a very beautiful slide of the thyroid gland, one of my favorites. So you can see the colloid has been stained pink. Very, very beautiful. And the esophagus also has the lining, epithelium, the mucosa, then the muscularis externa, inner circular, outer longitudinal muscles, or HME. And again, appendix, can now see how basophilic these regions look, and then how eosinophilic these outer portions also look, mostly muscles. Thank you very much. I hope this video helps.